Okay, this is the PowerPoint for cellular respiration. Even though the title says cellular respiration and photosynthesis, this is only going to be on cellular respiration. So obviously this unit is talking about metabolism. We already covered photosynthesis. Metabolism has two parts. When you did photosynthesis, we talked that is basically anabolism. When you're making or synthesizing glucose, or it says molecular structure, and that required energy in the form of ATP and NADPH, which was considered endergonic. Now we're going to talk about catabolism. Catabolism is the opposite. It's splitting glucose or breaking down a molecular structure. So we're going to make or release energy, which is called exergonic, and this is cellular respiration. Okay, so here are the basics. Here's a definition. Cellular respiration involves the conversion of energy in the chemical bonds of glucose to a different form of chemical energy. In this case, it's going to be ATP. ATP is huge. It's going to be used in energy transformations in the cell. It transfers energy through the transfer of phosphates. Cellular respiration is not the same thing as like respiration, like breathing. It's a cellular level. There are two types of respiration, anaerobic, anaerobic. In the first video, we're going to do aerobic respiration, or most of it. In the second video, we'll cover the anaerobic part. Anaerobic respiration does not require oxygen. It's found in all cells. All cells can do anaerobic respiration. When an organism has only anaerobic respiration, which means it cannot do aerobic, it cannot be very complex. It cannot be a very large organism. Thus, only unicellular organisms can be strictly anaerobic. An example of anaerobic respiration, which will be covered in the second video, is fermentation, and that's used to recycle NADH, which is a coenzyme we will need in cellular respiration. Aerobic respiration requires oxygen. It uses mitochondria, a complex membrane-bound cellular mechanism. In bacteria, or prokaryotes, aerobic respiration occurs in the cytoplasm and in membranes, even though they don't have a mitochondria. In eukaryotes, aerobic respiration usually involves the mitochondria. Here is the overall chemical equation for cellular respiration. You have, and there's no subscripts here, it's just C6H12O6 plus 6O2. Those are considered the reactants. Yields 6CO2 plus 6H2O plus our ATP. The 6CO2 plus 6H2O are our products. The aerobic part of cellular respiration is a redox reaction, which means we have reduction and oxidation occurring. Our glucose is going to be oxidized or broken down into carbon dioxide, and our oxygen is going to be reduced or made into water. In cellular respiration, there are four types of reactions that are going to occur. The first one is redox, which we just covered. The second one is dehydrogenation. This is when you have two protons and two electrons are going to be pulled from a substrate to a coenzyme. So NAD plus will make NADH, and FAD plus will make FADH2. This is called dehydrogenation, and basically a hydrogen is being pulled off of something in the reaction. Third one is decarboxylation. A carboxyl group is going to remove from something or a substrate to release carbon dioxide and make NADH or FADH2 with that hydrogen group. And the last type of reaction is called hydration, and that is just water being added to the reaction. Okay, this next picture shows how energy is released during cellular respiration. It's released from chemical sources via anaerobic or oxidative respiration in a series of small steps. It is not going to be just one big chunk of release. It is going to go step by step, small little steps of releasing energy. This slow, controlled release of the same amount of energy allows it to be released in a much more stable, usable form. Okay, there are four stages of aerobic respiration, and they're occurring in two parts of the cell. The first part of the cell that we're going to start at is in the cytoplasm, and that stage is called glycolysis. We will go through each of these in detail. Glucose is broken down to pyruvate by substrate phosphorylation. NADH and ATP are released. But again, that's not going to make sense to you right now because we haven't gone over it in detail. The second part of the cell, where cellular respiration or aerobic respiration is going to occur, is in the mitochondria. Stage 2, Stage 3, and Stage 4 occur there. In the second stage is when pyruvate is degraded to acetate and combined with coenzyme A. NADH and carbon dioxide are released. In Stage 3, which is the Krebs cycle, NADH, FADH2, carbon dioxide, and ATP are going to be released. And in the last stage, which is the electron transport chain and chemiosmotic phosphorylation, which you have learned in photosynthesis, is when we get NADH and FADH2 is going to fuel our making of our ATP. Here's a diagram showing the 4 stages of aerobic respiration. We're starting in the cytoplasm with glycolysis, the formation of acetyl coenzyme A, which occurs in the mitochondria. The Krebs cycle, or the citric acid cycle, is the 3rd stage, and the 4th stage is the ETC. You also see here the amount of energy that will be made in each of those stages, in stage 1, 3, and 4, with stage 4 being the most energy made of ATP. All right, before we get into cellular respiration, we need to review a mitochondria structure. Remember, it has a double membrane, an outer membrane, and an inner membrane. The inner one folds inward, and those folds are called cristae. So the cristae are located right there, those lines. The area surrounding the cristae is known as the matrix, and that's where the enzymes are. The coenzymes are there, the water's there, the phosphates, and any other molecules that are required for cellular respiration. The outer membrane of the mitochondria is permeable to most small molecules. They can get in. But the inner membrane permits the passage of only certain molecules, like pyruvate and ATP, so it's more picky. The proteins associated with the membranes are involved too. Okay, here's a diagram of the mitochondria, and I see my arrows are off. Obviously, the outer membrane is on the outer. The inner membrane line looks, and the inner membrane and the cristae is the red line. The matrix is in the middle, the light blue area, and then the intermembrane space is the green area. Okay. All right, so here we go. The first stage of cellular respiration, the aerobic part, is called glycolysis. Glyco means sugar, lysis means break. So glycolysis means the breaking part of sugar. It does not require oxygen. So it is an anaerobic step, which means all organisms can do glycolysis. It does, however, require ATP. Because we are breaking glucose, which is a sugar, those covalent bonds are extremely strong. We need ATP to break those bonds. It also requires enzymes for each step. All organisms can do glycolysis in their cytoplasm. Basically, what it's doing is it's taking glucose and making NADH, which is a coenzyme, and ATP. NADH is a coenzyme that carries high-energy hydrogens. We saw that in photosynthesis, but it was NADPH. It's the same thing. Okay, so here's the picture we saw earlier. We were talking about glycolysis right now. And glycolysis is a 6-carbon molecule, C6H12O6. So basically what we're going to do in 3 different steps, and we're not going to go into the details here, but there are 3 different steps. We're going to use this ATP, 2 of them precisely. ATP is going to break down, the phosphate's going to come off, and then that will break down to 2-ADP. And what we get is we're going to take that phosphate that got broken off and add it to glucose to make fructose 1,6-biphosphate. And you can see the phosphates there are added. And then immediately, that's unstable, and immediately that splits into glyceraldehyde phosphate, which we abbreviate G3P. So now we have a 6-carbon molecule broken down into 2 3-carbon molecules. Okay, after that, here's our G3P. We have 2 G3Ps. We're going to there's like 5 other steps, and you don't have to know the details for that. But what we're going to do is we're going to make some energy. It says we're going to further addition of phosphate to each of the 3-carbon carbohydrates occurs by NAD phosphate redox reaction. All 4 phosphates are removed to add them to ADP to make 4 ATPs. So basically it's kind of complicated, but what we're going to do is we're going to make some NADH, and we're going to make some ATP. And in the end, we're going to make 2 molecules of pyruvate, which are 3 carbons each. For this is very complicated. For this, for cellular respiration, what's important is what is going in, or what is required for each stage, and what is the end products of each stage. If you know that, then that is the key component, and we will see that at the end of each summary of each stage of cellular respiration. So what we did is, because we made 4 ATPs, right here, 2 plus 2 is 4. However, we used 2 earlier, so we have a net of 2 ATPs. All right, so here's some detail. I did give it to you in the PowerPoint, but and it got a little lined, a little off here. It's hard to read. But there are enzymes for every single step. You do not have to know all of these details. Again, this is showing all the chemistry of each of the steps here. This is taken from a different AP biology book. that describes what's happening if you're really into the chemistry. But again, all you need to know are the ins and outs and the locations of each of these stages. Another version of this is just getting to the G3P. Moving on, where we have 2,3 phosphoglycerates. And finally, with all of this, we finally, at the end, have 2 pyruvates. This is the structure of pyruvate right there. That's what it looks like. Okay, so like I promised, here is what you need to remember for glycolysis. The requirements. What is required for glycolysis? In order to split sugar, you have to have glucose. Glucose is required. You have to have 2 ATPs in order to break down the glucose. You need 4 ADPs laying around, and you need 2 NAD pluses laying around. What do we get out of glycolysis? We broke glucose down into 2 pyruvates. We used 2 ATPs, so we have 2 ADPs left. We used 4 ADPs and added phosphate to them, so we made 4 ATPs. We had 2 NAD pluses, and we added H's to them, so now we have 2 NADHs. Yes, you have to know the numbers, and along with the molecule names. The net result, though, however, because we used 2 ATPs, we have a net of 2 ATPs and 2 NADHs. We don't usually list pyruvate as a net result because pyruvate is going to move on to the next stage. Okay, so now we are finally going to move into our organelle, mitochondria. The mitochondria takes the output of glycolysis, which was pyruvate, and it's going to now be able to produce a lot more energy. Because remember, the mitochondria is the powerhouse of the cell, which means it's going to make ATP for the cell. So we are in stage 2, the formation of Acetyl Coenzyme A. All right, in stage 2, the formation of Acetyl Coenzyme A, which is abbreviated CoA. Pyruvate is going to enter the mitochondria. It goes right through the outer membrane. Oxidative decarboxylation occurs. Carboxylation means a carboxyl group is going to be removed from pyruvate. That carboxyl group, remember, is carbon double bonded to oxygen, OH. This is really CO2. So carbon dioxide actually gets released, and this hydrogen is going to be added to NAD plus to make NADH. It is oxidative. That term means that it's getting broken down. Pyruvate is getting broken down. So instead of having a 3-carbon molecule, we now have a 2-carbon molecule. The 2-carbon molecule is oxidized and the electrons are transferred to NAD+, creating NADH, which I described already. The remaining group, after all that occurs, is called an acetyl group. This fragment is going to attach to a coenzyme A, which is a molecule that's already there in the mitochondria, to create acetyl coenzyme A. So what is coenzyme A? Coenzyme A is actually vitamin B, and it is naturally made in cells. However, the bond between the acetyl group and the coenzyme A is very unstable, and it does not last for very long. Here's a diagram picture of stage 2, the formation of acetyl coenzyme A. So here's our pyruvate. We're going to take the carboxyl group right here off, which is called decarboxylation. Carbon dioxide is released, and that hydrogen that isn't really being shown there, it's right there. That hydrogen is going to add to NADH, or NAD plus, to make NADH. And then coenzyme A comes along and adds to that, and finally we have acetyl coenzyme A. That yellow bond right there is the unstable bond that is going to break soon. All right, so in summary, the requirements for the formation of acetyl coenzyme A is pyruvate, NAD+, and coenzyme A, which is vitamin B. The products are acetyl coenzyme A, carbon dioxide, and NADH. Now you can move on to video 2.